Welcome to topic 2.3 of module 2. In this particular video, we will be learning about osmosis and tonicity. The learning objectives for this topic are to describe osmosis and osmolarity, describe how 1 molar of sodium chloride provides approximately 2 osmoles in solution, describe what tonicity is and how osmosis relates to iso, hypo, and hypertonicity, and finally, to describe the role of plasma proteins in relation to capillary permeability and edema. Osmosis is the diffusion of water through a selectively permeable membrane. That is, from an area of high water concentration to an area of low water concentration. The selectively permeable membrane represents the plasma membrane of a cell. The plasma membrane is permeable to water. However, it is not very permeable to large molecules such as glucose, proteins, and ions such as sodium. To better understand osmosis, let's assume that we have a tank filled with water. The water in the tank is separated into 2 compartments, A and B, by a selectively permeable membrane. Now, because of the kinetic energy inside the water molecules, they constantly move around and collide with the permeable membrane, and eventually they pass through it. Because there is no solute in the water, equal amounts of water molecules pass through the membrane. Now let's assume that we make the water in compartment B a 20% sodium chloride solution, and the water in compartment A a 10% sodium chloride solution. Because there are a greater number of sodium and chloride ions in compartment B, these ions will try to diffuse into compartment A. However, because the membrane is not permeable to sodium chloride, it wouldn't allow the ions to pass through. Osmosis occurs whenever the water concentration differs on the 2 sides of the membrane. A very important thing to remember here is that if the solute concentration on the 2 sides of the membrane differs, then the water concentration differs as well. Because as the solute concentration increases, the water concentration must decrease. Due to this difference in the concentration of water, water molecules from compartment A pass through the permeable membrane and move into compartment B, eventually increasing water levels in compartment B. This movement of water continues until the concentration of water molecules is equal on both sides of the membrane. The following figure in this slide is a textbook example describing osmosis, where compartment A and B are separated by a selectively permeable membrane. Compartment A has a lesser number of solute molecules and a greater concentration of water molecules, whereas compartment B is the exact opposite, meaning it has fewer water molecules and more solute molecules. And because of this difference in concentration, the water molecules from compartment A pass through the permeable membrane and enter compartment B. This movement of water continues until the concentration of water molecules is equal in both compartments. Once again, diffusion is the movement of water from a solution of low solute concentration to a solution of high solute concentration. This is due to the fact that the solute molecules occupy more space, and a greater solute concentration means less water per unit volume. For example, osmosis drives water molecules from a 1% saline solution into a 2% saline solution across a semi-permeable membrane because of the difference in the concentrations of water between these 2 solutions. The tendency of water to move into the cell by osmosis is called osmotic pressure. Each cell in our body has a limited capacity to take water by diffusion. Once the cell is at its limit and cannot take any more water, it exerts a back pressure that pushes the water against the membrane or out of the cell. This is called hydrostatic pressure. At this point, there is no further intake of water. The hydrostatic pressure of a cell is equal to its osmotic pressure. As discussed in the previous video, water moves into the cell via gaps or special channels in the lipid bilayer called aquaporins. Aquaporins are integral proteins with tiny channels that only allow water to pass through them. Penocytosis, or fluid phase endocytosis, is another process by which water is moved into the cell, which is essentially ingestion of water into a membrane-bound structure before transporting it into the cell. Osmolarity is a measure of the total number of solute particles in a solution. Knowing the osmolarity, or number of solute particles in a solution, is key to how much water it can displace or move through osmosis, as one molecule of solute can displace one molecule of water. Osmolarity itself is directly proportional to the solute concentration. The unit of osmolarity is the osmol, or the mini-osmol. The osmolarity of our intracellular fluid is around 300 milliosmoles. Seawater, however, is 4 times more concentrated than the intracellular fluid. Therefore, the osmolarity of seawater is around 1,200 milliosmoles. The osmolarity of a solution depends on the substrate within it. For example, glucose does not dissociate into smaller particles. Therefore, the osmolarity of 1 molar glucose is 1 osmol. Whereas salts, such as sodium chloride, when dissolved in solution, tend to dissociate into 2 different particles, sodium and chloride ions. Because of this separation, sodium chloride has an osmolarity of approximately 2 osmoles. However, occasionally the sodium and chloride ions may bump into each other and exist as sodium chloride for a very short period of time. As a result of this, the actual osmolarity of sodium chloride is closer to 1.8 osmoles. Tonicity is the ability of a solution to change the shape and volume of a cell by changing its internal water concentration. Many solutes, particularly intracellular proteins and selected ions, cannot diffuse through the plasma membrane. Depending on the solute concentration of a solution surrounding the cell, cells can gain or lose water through osmosis. This changes their shape and volume. It's important that cells maintain their shape and volume in order to function properly. Isotonic solutions have equal numbers of solute particles in the intracellular fluid as outside the cell. Cells exposed to isotonic solutions retain their normal shape and exhibit no net loss or gain of water. The extracellular fluid, and most solutions that are given intravenously, such as normal saline, are all isotonic. Hypertonic solutions have a higher concentration of solute particles in the solution outside the cell. Cells immersed in hypertonic solutions lose water and shrink because water moves from the inside of the cell to the outside of the cell through osmosis. Hypotonic solutions are more diluted than the intracellular fluid, which means the concentration of solute particles is greater inside the cell. Therefore, water from the solution outside the cell will move into the cell, increasing the cell volume until it bursts. In capillaries, osmosis occurs through gaps between the cells, but not through the cells lining the capillaries. Blood plasma is sent everywhere to serve cells with nutrients and respiratory gases. The osmolarity of the blood determines the interstitial osmolarity, which in turn determines the cell volume. Different molecules take different routes to diffuse through the capillaries. Lipid-soluble molecules, such as respiratory gases, diffuse through the lipid bilayer of the endothelial cells. Ions, such as sodium and potassium, move through special channels between the cells that can be opened and closed. And finally, large molecules, such as proteins, are transported via vesicular transport. Capillary permeability is the capacity of a blood vessel to allow the flow of substances in and out of the vessel. Capillaries are lined by a single layer of endothelial cells that are connected by intercellular tight junctions. These junctions are permeable to water and ions. However, they are too small to allow proteins and other large molecules to pass through. Capillary permeability can differ between tissues. For example, in the liver, proteins and other large molecules can freely move in and out of the capillaries, as they have larger junctions through which they can pass. On the other hand, the capillaries inside our brain are impermeable to most substances, which is why treating CNS infections with antibiotics can be really difficult, as the medication often cannot reach the target tissue within the nervous system. Colloidal osmotic pressure, also known as oncotic pressure, is the force created by large, non-diffusible molecules, such as plasma proteins, that pull water back into the circulatory system. Plasma proteins inside the capillaries are too large to cross the capillary wall. Therefore, these molecules draw water towards themselves. In cells, proteins are also too large to cross the intact lipid membranes. For this reason, we call these proteins non-penetrating solutes. These can only be transported in or out of a cell using protein transporters via facilitated diffusion. High intracellular protein concentrations generates a positive intracellular pressure because they cannot pass through the plasma membrane and have the ability to attract water by osmosis, drawing water into the cell. The blood contains a large amount of proteins in the plasma that cannot cross the capillary wall. These proteins have the ability to attract water into the circulatory system by osmosis. As a result, we name this force the capillary osmotic pressure. Plasma capillary osmotic pressure is important in regulating the water distribution between the different extracellular fluid compartments, both plasma and interstitial fluid. In cases where there is increased water movement into the interstitial fluid, edema of the tissue occurs. Edema is an abnormal increase in the amount of interstitial fluid in a region of tissue. Malnutrition and liver failure are both conditions associated with low levels of plasma proteins. As these plasma proteins are largely responsible for drawing water back into the circulatory system from the interstitial fluid, low plasma protein concentration leads to swelling or tissue edema. During inflammation, the inflammatory chemical histamine increases capillary permeability by expanding the intercellular junctions. This allows plasma proteins to leak into the interstitial fluid, thereby increasing its osmolarity. As a result, large amounts of fluid is drawn from the capillaries into the interstitial space, leading to edema. In this video, we have learnt about osmosis, osmolarity, isotonic, hypotonic, and hypertonic solutions and their ability to change the shape and volume of a cell, capillary permeability and colloidal osmotic pressure, and the different causes for edema.